This program is a presentation of UCTV for educational and non-commercial use only.
Welcome to the second of the Hitchcock Lecture Series. I'm Fan-Yong Liu, a professor from School of Public Health, and also the chair of the Graduate Group of Compared Biochemistry. On behalf of the Dean of the Graduate Division, as well as the Hitchcock Selection Committee, it's my great pleasure as well as my honor to introduce today's speaker, Professor Sidney Altman. Professor Altman is truly a distinguished scientist and an outstanding teacher. More importantly, he's also a wonderful mentor, as I personally experienced as a postdoc fellow in his laboratory at Yale. I will not repeat the long list of his accomplishments here as a Professor Lester did yesterday. However, I would only want to add that he has spent his entire 40-plus years career focused on a single yet critically important enzyme called Ribonuclease P, or RNase P. His scientific journey on RNase P is an inspiring story. It starts with his initial discovery of the substrate as well as the enzymatic activity of the enzyme RNase P. When he was a postdoc fellow, under the guidance of Sidney Brenner and Francis Crick at MRC Laboratory of Molecular Biology in Cambridge, England. He then moved to Yale, subsequently cloning the RNA subunit of the enzyme RNase P and demonstrating the catalytic activity of the RNA subunit. His research has revolutionized the field of molecular biology, specifically the function of RNA molecules. Today, he will give us his personal reflection of the discovery of the catalytic RNA and share with us his recent research progress in his laboratory. And please join me in welcoming Professor Altman.
Thank you very much, Fan-Yong, for the introduction and for your wonderful hospitality during my visit here. And once again, I want to thank the members of the Hitchcock Committee for selecting me for these lectures. And I hope I deliver in some reasonable way. I want to start by repeating something I said at the beginning of yesterday's lecture, which is that you've all paid for my research over the past 30 or 40 years. Your income tax, of which a few pennies on a dollar, so to speak, are devoted to my particular research through the National Institutes of Health and the National Science Foundation. And I want to thank all of you taxpayers for your donations over the years. I feel that's a way of indicating my appreciation instead of showing a slide with many different names and many different agencies on it. Okay, so yesterday we talked about the origin of life a little bit and what the RNA world was. And today I'm going to focus on ribonuclease P, which Fan-Yong indicated, which has a subunit that is catalytic. And what I'm going to do is talk about both the RNA and the protein subunits of RNase P in various organisms. And I want to indicate that RNase P is an essential enzyme in all organisms. I won't go through the data, but it is clear that without RNase P, these organisms don't grow. And after I talk a little bit about the enzyme, I'm going to talk about different substrates, both in E. coli and in higher organisms. And then the last part of my lecture will be on some recent experiments in my lab in which RNase P is an important mechanism in providing a drug therapy. The first slide you see is an old slide, and it indicates the first substrate for RNase P. This is a tRNA molecule, as I showed you yesterday. It's in 2 dimensions. And these are extranucleotides at one side of it. And RNase P cuts right here at nucleotide 1 of the mature tRNA sequence. So all these nucleotides are removed. These nucleotides are extranucleotides too, and they're removed by different enzymes. So essentially, we make virtually a functional tRNA out of this cleavage here. One of the problems we had to confront, and I think we solved it as I'll show you later, is what is the specificity of this enzyme? In fact, there are about 60 different tRNA precursors in every cell. None of them have a consensus sequence or similar sequences around the cleavage site. And so how does this enzyme recognize its substrate? It does recognize it by looking at a partial RNA helix on this side of the substrate and a single-stranded region on this side here. We have 2 extra base pairs here, but it's not at all clear that they form in vivo under the conditions that we're talking about. Okay. Now, this is a chart which characterizes the catalytic activity of the RNA subunit of RNase P. Let me just say, although you cannot see it clearly, it's not shown here, the structure of the catalytic subunit, although I will show a picture of that as it has been recently crystallized by Mondragon at Northwestern University. The catalytic structure is extremely similar in all organisms. However, there is one difference between that and the proteins in the enzyme. In E. coli bacteria, let's say in E. coli and in bacteria generally, there's one RNA subunit, which is catalytic, and there's one very small protein subunit. And if we can rate the catalytic activity in bacteria, let's say it's of the order of 1 to 10 here. Then we have other species involved, the archaea, which we now classify as a separate kingdom here, and the eukaryotes. In archaea, there's one RNA subunit and 4, and possibly some cases more, proteins in the enzyme. The RNA subunit is catalytic, in several cases tested, but the catalytic activity is only about 1/10 to 1/100th of that compared to that in E. coli, but it's easily recognizable. And this is at pH 6, where in fact there's very little degradation of the RNA as you incubate it in a reaction mixture, so that you can run reaction times overnight at pH 6 under the conditions Leif Kirsebaum in Sweden really devised, and you can easily see very low amounts of activity. And Kirsebaum and his group found that the RNA from a couple of eukarya there's one RNA subunit, and there are 9 or more protein subunits, and the catalytic activity here is about 10 to the minus 5 compared to the catalytic activity here. So these are measurements that have been made rather carefully, so we're very confident of these numbers. This indicates the uncatalyzed reaction, so if you just let the substrate sit around, you'll find the cleavage rate is around 10 to the minus 9 or so. And here are the relative references. Guerrier Takata et al. In E. coli, Panucci et al. In at least one of the archaea, and Kikovska and Kirsebaum et al. On eukaryotes. I think that's all I'm going to say about the RNA at the moment. Now we're going to talk about diversity in the subunit composition of RNase P. So we have in bacteria one protein, which we had labeled a long time ago, C5 protein because of just of its position in gels. In archaea, there are 4 different proteins, and now another one has been characterized, which is actually a ribosomal subunit, which, when added to these 4, stimulates the activity considerably. And it has some homology with one of the proteins found in yeast and in HeLa cells. And we have in HeLa cells, this is a human enzyme, 10 subunits here, and you can see some of them have homology to the archaea subunits. Here's RPP30 here, RPP38 here, et cetera, RPP21 and 29 and POP5 here. But there are several subunits here, and as I indicated from the previous slide, they appear to contribute to the catalytic activity. And by saying appear to contribute, it could very well mean stimulating the RNA activity by itself by adding these various proteins. So I should also say something about the evolution of the RNA subunit. The shape, as you can draw it in at least 2 dimensions, is very similar from bacteria up to human cells. There are some differences. Generally, it's about, the RNA is about 350 nucleotides in size, but there are a couple of yeasts where it's only 250 nucleotides in size, and there are a couple of other organisms where it's like 800 or 900 nucleotides in size. However, the difference in size corresponds to a difference in the recognition of certain subunits and does not seem to affect generally what we observe. The catalytic portion of the enzyme remains more or less constant, although does change somewhat between the eukarya and bacteria. And these slides are from Venkat Gopalan at Ohio State University. And this gives an indication of the evolution of the protein subunits. We have a common ancestor here, where presumably RNase P lacked protein subunits. And although there's no direct evidence, there's nothing to indicate that that should not be the case. We know that if we isolate the RNA subunit by itself in test tubes in vitro, it has the catalytic activity that is for bacteria, and we can show it for these other organisms. And then for bacteria, there's the origin of a single protein cofactor. And for the other organisms here, apparently you start sharing 4 protein subunits, and then you have the archaea, and the eukarya are out here. So this is a rough picture of the evolution of this enzyme in terms, if I may make the reference to what I talked about yesterday, this is an indication of the complexity of evolution as it goes along. So let's not think about humans as thinking individuals for a moment, but if we just look at the structure of a key enzyme in this case and determine what goes on, the bacteria are very simple, but eukaryotes are quite complicated in this particular case, and we see that the proteins seem to have replaced the contribution of RNA in terms of the catalytic activity. Now I'm going to talk about some substrates that have been found in E. coli to date, and some of them exist in other bacteria as well. First, we have tRNA precursors, which are listed here, and as I said, there are about 60 different precursors in any particular organism, bacteria or eukaryotes. Then we have 4.5 sRNA, in which the structure can be drawn like this, so long double-stranded regions, long hairpin. But the precursor has extra nucleotides here, and the arrow indicates where E. coli will cleave it, just at this point here. So this, in fact, was good reason to believe that our other data on the structural features that are required for cleavage by RNase P is correct. That is to say, we have a long period of double-stranded RNA here, of which actually you only need about a half a helical strand of a helical twist of RNA, and a single-stranded region here. This is another similar substrate from E. coli. Here are others. I won't go into what they're doing at the moment. This is a phage RNA, actually. This is actually tmRNA, or an RNA which is both a messenger RNA and looks like a tRNA. That is to say, there's a coding region in this region here, and it codes for a small protein of about 11 amino acids, which tags proteins that are to be degraded inside E. coli and other organisms. But the ends of the molecule look just like a tRNA molecule, and it has a precursor sequence here, and RNA cleaves it right here. So these are all important molecules in E. coli, and they're not unstable molecules. They have reasonable lifetimes inside cells, and if you know how to look for them, and now we know how to look for these, you can find them quite easily. Several years ago, about 7 or 8 years ago, in my lab it was found that there was another set of substrates for RNase P, and it remains to be shown by really rigorous experiments that this does function in gene regulation. But we've shown in model systems that it does. So here are various operons indicated. An operon is a region that has 3 genes, usually coding for a similar metabolic function, but not always. Here's another one here with 5 genes. Here's the histidine operon with several genes. And the fat arrows are coding regions for proteins. And these narrow lines here are the regions between proteins. And what we can say, for example, is that RNase P in these cases cleaves in the regions between the coding regions. So these underlines indicate cleavage here, one here, and one in this region right here. We isolated these molecules just by looking at a temperature-sensitive mutant in RNase P at a high temperature, and we picked these molecules out from many molecules that you could find under these conditions that did not appear under normal growth conditions. So these are some examples of cleavage in an operon, but it's much easier to do things in an operon that you really recognize, which is the lac operon for the metabolism of lactose. And here we have the lac operon. There are 3 genes, LAC-Z, LAC-Y, LAC-A, and we know what each one of these codes for, and here are the intergenic regions right here. So here's the end of the LAC-Y region in this particular case. So we're looking at this region in here, and this is the beginning of the LAC-A operon, a UUG. This is a UAA here. And this is what the region between the 2 coding proteins looks like. And it looks like it actually has a structure that would be recognized RNase P, and in fact it does. This is where P cleaves, it's right here. And I won't go into data, but this just indicates what I'm talking about. That is to say, if we put this structure on a piece of genetic information where it's coded for in a bacterium where we can turn off RNase P when we want to, and we can do that, we show that when we turn this off, we make more of LAC-A. When we allow P to function, we make much less of LAC-A because you get cleavage here and degradation under these cases. Now, it was noticed in the 60s when RNase P, when the LAC-Z operon was being studied by many people, that there was a significant difference between the translation or the production of these 3 genes here. In fact, there was much less of LAC-A made than LAC-Y or LAC-Z, and there was really no explanation at all given for that. And I think in this case, with the model system, we have actually supplied that explanation. But this simply gives you another indicator of the range and variety of substrates for RNase P in bacteria. So we can talk a little bit about now in eukaryotes, and David Spector some time ago published a paper in RNA. I think it was 2000, it was either early in 2010. Yes, it was. And he asked us for samples of RNase P from HeLa cells, which we sent to him. And he was looking at a substrate that looked like this. We don't have to worry about most of this. This is just taken from his paper. In fact, he was looking at a very long piece of messenger RNA, and inside the message, he identified what looked like a tRNA molecule here. And as it turns out, RNase P does cleave this molecule right at the junction of the single and double-stranded region here, and you wind up with this. Okay? This describes further events in the pathway, which I'm not interested in for our purposes at the moment. But he clearly had found that RNase P does cut in that particular position, and he recently told me that he has at least one other such case of substrates that look like this in eukaryotes. So that's satisfying. We already knew from previous experiments, of course, that RNase P cut tRNA precursors, for example, in HeLa cells or mouse cells or any other organism that you wanted to look at. And we know that RNase P is actually located in the nucleus of eukarya, and I might mention that a little later. So it cleaves tRNA precursors, it cleaves these long messages with tRNAs in the middle of them, and we have evidence, and we've published evidence, that it cleaves ribosomal RNA precursors in HeLa cells and in yeast. It's hard to get purified ribosomal precursors, but the evidence we have is pretty convincing. So we have at least 3 classes of substrates in eukaryotes, and I'm sure there are more substrates. It's just that we haven't, and I don't think anybody has made the effort to look for more. Now, it had been suggested for some time that in various organelles in eukaryotes, for example, mitochondria or chloroplasts, for example, that there was no RNase P of the kind that I've mentioned. It had been indicated there was RNase P that contained no RNA at all. However, a recent paper by Kohler and Teitel in Cell this year, a few months ago, it actually was quite a remarkable paper. It was on the function of an enzyme, polynucleotide phosphorylase, PNPase, which is responsible for importing various pieces of RNA into mitochondria in human cells. And one of the things it imports is RNase P. And in fact, Teitel and Kohler and their co-workers showed that there were certain sequences that actually were cleaved by the conventional RNase P. Now, these diagrams are taken from another paper, but it doesn't really matter. They demonstrate what I wanted to show. And that is, when you have the classical RNase P with an RNA subunit, and in mitochondria you have situations where you have a long piece of RNA, and interior in the RNA there might be 2 tRNA sequences. You have messenger RNA here, messenger RNA here, and in these kinds of cases, RNase P cuts it here, the beginning of this tRNA, and cuts it here, the beginning of this tRNA. It had been shown by people who worked on these enzymes that supposedly don't have RNA associated with them, that if you have an RNA like this at the head of a region where there's a messenger RNA here and you have one RNA here, that these other enzymes cleave this RNA at this particular point. So that is a remarkable new finding. I found that the whole paper that I looked at in this case was a very interesting paper and very useful, and it showed, in fact, that RNase P seems to be in quotations everywhere, a classical RNase P. There's one other thing I'd like to show you about the structure of the enzyme, and it's complicated. It's essentially the crystal structure, a fantastic crystal structure. performed by Mondragon and his colleagues at Northwestern University. They have solved the crystal structure of the ternary complex, which contains the RNA subunit of RNase P from a bacterium that's not too different from E. coli, the protein subunit of the complex, the tRNA product of the complex, and a single-stranded piece of RNA which supposedly would represent the cleavage product from a tRNA precursor. So effectively, we're looking at the product of the reaction with the product in the substrate. This paper will be published in Nature soon. I can effectively guarantee that. Okay, so this is a different way of looking at it from what the conventional picture is, but here we have in blue or purple, whatever you want to call it, the RNase P RNA. And it's organized in this particular way. Usually we turn it at least by 90 degrees and look at it from that point of view, but that's all right. And I'm not going to go into any of the crystallographic details at the moment. I simply want to show that this is what has been found and how it confirms some of the predictions that were earlier made. This is the tRNA precursor here. So this is the anticodon stem down here. And this is the recognition site here at the junction of the tRNA molecule. And there would be a single-stranded region which we can't identify here. And the protein cofactor is located in this region here. Now, there has been a lot of RNA work done on this before the crystal structure. So we already knew something about which parts of the RNA were in contact with the protein. We knew by cross-linking studies which part of the tRNA were in contact with this piece of the RNA too. Nevertheless, I think this is a fantastic piece of crystallography and very interesting work. So I offer my congratulations to Mondragon. We had shown that the RNase P in complex with a substrate has different conformations. It can be the transient state or the active state of the enzyme, and we don't know anything about that yet. It could be the free substrate, or it could be the cleaved product and the single-stranded region. But we had shown from various enzymological experiments in vitro that these three sites, these three conformations must exist, and Mondragon has succeeded in producing the crystal structure of this one. We had also shown from cross-linking and other studies that the aminoacyl helix of the substrate is denatured during the cleavage product. That is to say, at the helical end of the tRNA precursor, where usually at the 3' end an amino acid is attached, and the 5' end you have an extra substrate, those strands come apart, so it's no longer a double-stranded region in the helix. And we'd shown that that was the case, and if you cross-link the helix and it could not denature, then you got no enzymatic reaction at all. And that was confirmed by this particular structure. It was shown by Mondragon that, in fact, the two parts of this helix are denatured in the complex. And furthermore, the position of the protein has been specified by various methods, and the crystal structure confirmed all of that too. So it's quite satisfying. Okay, I'm going to go on now to some recent work in our lab, and I might skip some stuff because I don't want to go over time. First of all, here's the classical tRNA precursor. And it was from these molecules that, in fact, we first deleted this region here, this stem and loop, and this stem and loop. It was done with Bill McLean, and we covalently linked this to this. And that was a very good substrate. It was just one continuous helix with a loop. And then Tony Forster, a postdoc from Australia, this is 15 years ago at least, succeeded in deleting this loop here, So we're just left with this strand, hydrogen bonded to this strand here. And if you draw that differently, this is what you see. In fact, these are now two different RNA molecules, but they get cleaved very well by RNase P at this point here. And they've been drawn in different colors here, but because now they represent the basis of the therapy I'm going to talk about. This can be any target RNA in a cell, any cell, okay? And as long as you have another piece of RNA that can hydrogen bond to it here, this is especially true of bacteria in this case, we call this the external guide sequence, the EGS. And as long as this can hydrogen bond here, this is still a very good substrate. It's just an imitation of model substrates we made, and it's cleaved by RNase P. Now, if we have a piece of DNA, which is made into an RNA or messenger RNA, whatever you want, and there's no external guide sequence involved, you make protein, the regular state of affairs. If you have an external guide sequence, hydrogen bonded to the piece of message, then cleavage is going to occur at one particular end of the EGS, and you're going to get mRNA cleavage there by the host cell RNase P, which recognizes this is a substrate, and you're going to inactivate this particular RNA, and you're going to stop it from expressing itself completely. So if the RNA is a viral RNA, or an RNA that codes for a particular disease, you're going to absolutely stop it from functioning under those conditions. So that's the basis of the therapy, that you have this situation, and you have the RNase P, which exists inside the cell, and so you can do all these things. Now, these are some ways of transporting any piece of RNA. There are various other mechanisms in which RNA is involved, and I'm not going to talk about them at the moment. Let me just say that the method I'm going to describe is just as good as siRNA. We've tested siRNA on the same targets as we've tested the EGS on, and the numbers are very comparable. You can have things attached to antibodies, which themselves attach to particular parts of cells. You can have them attached or inserted into viruses, which then infect cells. You can have oligonucleotides encompassed in liposomes, which are lipid bodies that will wrap around a piece of RNA, or a nanoparticle containing some RNA, or you can have basic peptides attached to pieces of RNA. Although our previous work had been done biologically, that is to say, we made small genes that coded for EGSs and inserted them into bacteria, and they certainly work in E. coli and some other organisms. But now we're doing these experiments with basic peptides with Morpholino oligonucleotides, and I'll show you a picture of that very shortly. And this is just an indication of the kinds of RNA you could use to shut down gene expression. There are aptamers, which are small molecules that bind to particular sequences. So these are made out of RNA, let us say. Hammerhead ribozymes, which are not very good, I think. siRNA, which can be used in human cells, for example. Splicing variants, which have been used to attack certain diseases in which there is a splicing variant that causes diseases like Duchenne's muscular dystrophy, and you can adjust that splicing variance back to the normal situation, and in a couple of papers which have been published, you get some improvement in the muscular dystrophy, the EGS, which is what I'm going to talk about, locked nuclear RNA, which is another variant on RNA, and peptide nuclear RNA. I'm not going to talk much about these, but they can be useful. This is an old slide showing results with our biological effects. Simply that in E. coli, for example, we can inhibit the expression of various of these genes When you induce beta-galactosidase or alkaline phosphatase, they're made at a level of over 1,000-fold of the constitutive level. So normally they're hardly made at all, but you can induce them, and then the level goes up. With one EGS, attacking one site in the message, you knock out 60% of the activity. These are essential genes, but this one is the one that we're interested in, drug resistance. We're looking essentially at chloramphenicol resistance, and we can stop that, we can reverse that into chloramphenicol sensitivity of a bacteria 100% by using two EGSs that attack two different sites in the messenger RNA that codes for chloramphenicol resistance. Okay, you can say that's all right, but conventional antibiotics are just as good. Well, they're not just as good. A one-step mutation can convert something that's sensitive to drug resistance. In our case, 3 mutations, as long as they're not right next to each other in the EGS, can still function. So it's clear that that is a much better biological result than the conventional method of doing things. And this has also been replicated by somebody else actually in California, now that I'm here, on amikacin resistance. Just let me go on now. Very briefly, I will say that we can now isolate an EGS in a few hours, providing you have a supply of E. coli RNase P on hand, which is very easy to arrange, and a half mil solution of a piece of random RNA. So in our case, we might have something of this long here, where N is the random RNA in these pieces, and we have various restriction sites here. And we just incubate this. You can label the target RNA in any particular RNA you want. You can mix that labeled target RNA with this random oligo here, and then incubate it for 15 minutes with RNase P, and run it out on a gel. Takes 2 hours perhaps. Develop a film on the gel, and you will see immediately whether or not you had any cleavage of the target RNA. And in fact, we've shown this prolifically over the last couple of years using this method. We've identified EGSs in many different bacteria, and they can be used for drug sensitivity to target pathogenic regions in pathogenic RNA. It's a very convenient method. Now, let's get to this new method, which is the basic peptides attached to Morpholino oligos. This is a Morpholino oligo. You see the phosphate linkages are not quite what you would expect. There's a nitrogen in here. This is a Morpholino sugar in here, a nitrogen in it, but the bases appear as they usually appear. So, and the other aspect of using this is that the Morpholinos are tremendously resistant to non-specific nucleases. So you can incubate them with human serum or bacterial growth medium, and they don't get degraded for at least 24 hours, probably much longer than that. And that's why we use them. Now you attach a basic peptide at one end, and here it's 14 amino acid residues. R is arginine, X is 6-amino-hexanoic acid. So there are 4 repeats of this, X again, and then beta-alanine. And this makes the covalent linkage very powerful in terms of penetrating bacteria. We published this about 1.5 years ago or so. Let me just say that if we're looking at chloramphenicol resistance in this case, and we're using 2 EGSs, which have been made into Morpholino oligos with a basic peptide attached. These are control bacteria after 4 hours, a scrambled oligo of the Morpholino, so it's almost as good as the control. 2 separate EGSs and 2 EGSs together. So in this particular case, we're down around 1,000th of what we would have expected for the control bacteria, which is actually a very good number. So that's at 50 micromolar. When we do it with B. subtilis and activating the gyre gene in B. subtilis, we only need 5 micromolar of this, and we're down to about, in this case, only 0.2 of the viability of this. If we do this at 15 micromolar, we're down to about 1% of viability of the control. So that works extremely well. And let me just say, in an experiment that was finished a couple of weeks ago, I'll just show some rough data. We are now doing the experiment with a different basic peptide. It's a basic peptide that has been isolated from human cells that controls T cell regulation. And it's 22 amino acids long. It's written up here. It is also very basic, and we've attached that to a Morpholino oligo. And this is chloramphenicol resistance. So we're down in the level between 1,000th and a 10,000th viability here. These are 2 different preparations. This preparation is obviously much better, and that's what we're working on at the moment. And this is at 5 micromolar concentration of the particular compound that I worked on, that I told you about. So that's a summary of some of our recent data. I have some more data on some molecular biological aspects of the Morpholino oligos and how they interact with sites and message, but I'm not going to show that to you because it's getting late. And I'll just show you this one other experiment, which is an older summary of experiments in eukaryotic cells. And I'm sure Fenyang will recognize some of this. I'm not going to talk about Cerevisiae right now, but we can inhibit flu viruses in mouse or canine cells by attacking 2 different genes in the flu virus. Remember, the flu virus has 8 genes, and it also has 8 pieces of RNA in its genome. Each piece of RNA codes for a particular one of its genes. And by attacking both of these particular genes, we essentially can inactivate flu virus by 100%. But I indicate 95% because there's a 5% error in our methods. These are essential proteins in E. coli and HeLa cells. I won't talk about them more at the moment. But Fenyang Liu has been working for many years on cytomegalovirus in mice and in humans. And actually, these are old data which showing that he can inhibit these particular functions. But I believe that he's also been able to show the inhibition of live animals under these conditions. So, and also herpes virus, which he also did in our lab a long time ago. So I think this is a very promising method to use, and one that's certainly worth studying. It is not necessarily adding to our molecular biological knowledge of RNase P itself and what it's done, but it shows that RNase P can be used in very useful ways, in this case, to attack all kinds of disease. And with that, I shall leave you.
Sid, I'm curious how the basic peptide Morpholino penetrates into bacteria so efficiently. It's not the same kind of mechanism that basic peptides like TAT enter into mammalian cells. It must be something different.
I'm not sure it's very different at all, quite frankly.
TAT peptide penetrates into mammalian cells by first being internalized into an endosome, and from the endosome into the cytoplasm.
Say the second half of that sentence again.
The TAT peptide, basic peptide, penetrates into mammalian cells by first being internalized into an endosome, and then from there into the cytoplasm. That's not happening.
It's certainly not that.
Yeah.
That's right.
I'm curious about the toxicity and pharmacokinetics of these molecules.
I don't know anything about it because I haven't done any experiments with live animals. Mm-hmm. However, there are people who have done experiments with these kinds of compounds at Avi Biopharma, which is a company now located in outside of Seattle, and they have cured infected mice under these conditions, and the mice don't suffer at all under the conditions they use. They just survive for several weeks afterwards.
That's encouraging. Thanks.
When you showed the cleavage of some of the polycistronic messenger RNAs in E. coli, I assume that was an in vitro experiment.
No, we've done it both ways.
Because it was pretty inefficient. It was only a few % of the intact transcript that was converted to product. So how do you expect it to contribute to regulation?
There are two different slides there. One was of several different operons. That was done in vivo, okay? And then we did the lac operon, which was done in vitro and in vivo.
But it's only a few % of the message, even in vitro, where you, is it more efficient in vivo, the degree of cleavage of the intact transcript? It was, the full-length transcript persisted, 95% of it was still there after, more than an hour incubation.
Well, let me put it this way with a lac operon. We completely destroyed part of the lac operon in vivo. Okay?
So it's more
that was with a model system. Now, so, I don't know what you want to say about that. With the other operons, it is true that we don't have a good idea of the quantitative result of those experiments.
Okay.
So does the bacterial protein stimulate the RNA of archaea and so forth? In other words, if you combine the different proteins and RNAs, are the proteins still active?
Which proteins?
The RNase P protein. So immediately, does the RNase P protein from E. coli, for example, stimulate the RNA of RNase P from archaea and so forth?
Well, if you're talking about experiments using an EGS to...
No, this is going back to the beginning of your talk.
Well, just under normal conditions?
Yeah.
Yeah. The amount of the P protein for RNase P, there are about 400 to 500 copies in the cell. And if you try and activate RNase P by attacking the message for the P protein, which we have done, it actually takes several more hours to kill those bacteria than it does, for example, to just change chloramphenicol resistance and sensitivity, because you have to deplete that reservoir until you're down to maybe 20 molecules per cell, which is apparently not enough to keep the molecules going.
EGS, what needs to be done to bring EGS as a therapeutic technology into the industry?
Somebody has to provide a few $1 million to set up a company to investigate this for commercial purposes and to build a reasonable system for delivering the covalent linkage of the basic peptide to the morpholino. So I don't know anybody with a few $1 million who's willing to do that, and I'm not a great entrepreneur, so I can't do anything about it.
And a follow-up question. Is it easier to design an EGS for viruses or as opposed to cancers?
In principle, you could, certainly for viruses. And that's what Fenyang is doing.
Well, I want to follow Stu's question. How much we know about the biogenesis of the enzyme in terms of the RNA subunit and the protein subunit in E. coli?
Just a minute. Could you stand a little bit further back from the microphone? Not that far back, okay.
So how much we know about the biogenesis of the RNA and protein subunit in E. coli? I mean, specifically when the bacteria grow under different conditions, like under stress or under rich medium. Do we see any...
For the protein?
As well as the RNA subunit.
Okay.
Is there any regulation?
The protein is made in an operon with some ribosomal proteins in it. And supposedly there is a binding, the sequence of the upstream part of that operon as a binding site for the RNA subunits. But we've done a number of experiments in which we've tried to regulate the amount of RNA and the protein in E. coli, and none of them have worked. That is to say, changing the amount of RNA doesn't change the amount of protein, changing the amount of protein doesn't change the amount of RNA. That's about all I can say about that. Is that what you wanted to know?
Because I'm wondering whether the tRNA synthesis is affected by
that's
the...
a separate issue. The RNA subunit is under the relaxed stringent control. Okay? And I think so is the protein subunit. So if you starve the cells for amino acids, or they go into stationary phase, you stop making both the RNA and the protein subunit.
Please join me in thanking Professor Altman for a wonderful lecture today.